Hi everyone. Now we are going to discuss about amoebiasis, which is one of the type of food and waterborne diseases. Amoebiasis is caused by a protozoan parasite called as Entamoeba histolytica. This Entamoeba histolytica is a very common parasite which is going to cause endemic in warm climates where adequate sanitation and effective personal hygiene is lacking. About 90% of infections are asymptomatic. That means the amoebiasis that was caused by Entamoeba histolytica, in about 90% of infections are asymptomatic. They won't show any symptoms at all. But remaining 10% is going to produce a spectrum of clinical syndromes ranging from asymptomatic to dysentery to abscesses of liver or other organs. That we will discuss in detail in coming slides. Coming to the history, Mr. Lambel in 1859 discovered this amoeba. In 1875, Loge a Russian differentiated that the amoebic dysentery from bacillary dysentery, that is E. coli, by describing amoeba in the stool. That means its pathological nature was discovered by Mr. Loge. Then in 1881, the councilman and Koffler described the true bowel lesions and used the term amoebic dysentery. Then Cartelius in 1887 found amoeba in the pus from a liver abscesses. Then in 1903, Squadin differentiated pathogen and non-pathogenic types of amoeba. That means the detailed study of amoeba was done by Mr. Squadin in 1903. This is all about the history of Entamoeba histolytica. Then coming to the causative organism of amoebiasis, the Entamoeba histolytica, the causal organism of amoebiasis, is a microscopic amoeba. Where it lives? It lives in the submucous coat of large intestine and it is a monogenetic. What is meant by monogenetic? It is going to complete its life cycle in only one host, that too mainly in the humans, and rest of the other animals are going to act as a reservoir, but it is not going to show its life cycle. Okay. Then moving to the morphology of that organism, that is Entamoeba histolytica. As I told you, it lives in the submucous coat of large intestine. And this Entamoeba histolytica parasite is going to exist mainly in the three forms. What are those three forms? One is the trophozoite form, which is also called as magna form or vegetative form. The second one is the precystic form or minuta form. And the third one is the cystic form. So how many forms of Entamoeba histolytica parasite is going to exist? Three forms. One is the trophozoite or magna form. The second one is a precystic or minuta form. And the third one is the cystic form. Let's discuss all these forms in detail now. The first one, trophozoite or magna form, which is also called as a vegetative stage of Entamoeba histolytica. So if you see here, you can see it is going to have an irregular shape. That means there is no proper shape without having the amoeba features, that means possessing of a pseudopodia. Okay, it is most active motile form with a pseudopodia. It lives in the mucus and submucous layer of the large intestine of man, and it is going to measure about 20 to 30 micrometers in diameter. The outermost body covering or plasma lemma, so that means the outermost covering of this trophozoite is going to be called as a plasma lemma, which is very thin, elastic and semi-permeable membrane. So because of this elastic nature only, it is able to form the pseudopodia. Now the protoplasm is differentiated into outer ectoplasm. So here you can see this is ectoplasm, and inner it is going to be called as a endoplasm. Outer translucent ectoplasm, here you can see, and inner is going to be glandular endoplasm. And if you see here, the RBC and WBC and some sort of tissue debris are also present in this endoplasm. And at the center, you are able to see the echocentric, which is nothing but the nucleus, which is spherical in shape, isn't it? Contains a central karyosome. So here it is, central karyosome, surrounded by a clear halo. And the nucleus is lined with a delicate nuclear membrane, and the karyosome is occupied by fine thread-like chromatin network with spoke-like radial arrangement. So here, this is all the things that you can see here. And here the trophozoite is going to be multiplied by binary fission. That means one cell becomes two, two becomes four. This is going to be called as binary fission. So this is all about the first stage or first form, that is going to be called as trophozoite or magna form. What is the second form? That is precystic or minuta form. So here you can see. So here some trophozoites undergo encystment in intestinal lumen. Now this cyst formation does not take place in tissues and in feces outside the body. Then where it is occurring? Only in the intestinal lumen, this precystic form or minuta form is going to occur. So before the encystment, the parasite becomes slightly ovoid. So here you can see the ovoid with a blunt pseudopodium. Okay, and the endoplasm is free of RBC. So where in the previous slide you can see here the RBC, WBC and the cell debris are present, but in the cystic form those are all are absent. Nucleus will be similar to that of the trophozoite, that means the karyosome, fine thread chromatins, all those are present, and it is going to be of very small, spherical, non-motile, non-feeding form, and it is going to measure about 12 to 15 in diameter, micrometers in diameter, and it is smaller in size, and the food vacuoles are absent. So then where it is going to live? Only in the lumen of intestine, and it is non-pathogenic to man. So this stage it is not going to cause any harm. Then moving to the third form, that is a cystic form. So under normal conditions, so here you can see the cystic form. Under normal conditions, the parasite, that is a minuta form, becomes spherical. So here you can see it's a spherical completely, whereas the previous one is going to be the ovoid. The parasite minuta form becomes spherical and secretes a refractile wall surrounding it known as a cyst wall, and this process is called as encystation. The process of forming this wall around this form, minuta form, is going to be called as encystation. Now this mature cyst is a spherical and measures about 10 to 20 micrometers in diameter. Its cytoplasm is clear and contains one or two glycogen masses which are going to act as a reserve food. So here you can see one or two. So if it is one, we call it as uninucleated cyst. If it is going to be of two, it is going to be of binucleated cyst. Along that, here you can see some sort of a bar-like chromatid bodies or chromatial bodies with rounded ends. Both glycogen masses and chromatid bars gradually disappear. Okay. And early cyst contains, as I told you, only one nucleus, that is single nucleus, where we call it as uninucleated cyst, but soon divides into two, that is going to be called as binucleated cyst, and then you can see four nucleus is there, which we are going to call it as quadrinucleate cyst. So that's how it is going on dividing its nucleus and finally forming a quadrinucleated cyst. A mature cyst is quadrinucleated and contains of masses of glycogen and chromatid bodies which are oblong, refractile and with rounded ends. So this is the actual picture of iodine mounted cyst, where you can see the nucleated karyosome, and here is the chromatid bodies and some sort of the glycogen masses. So this is a real picture that of this cystic form. Now we have learned all the three forms of Entamoeba histolytica, that is trophozoite. Then the second one is the minuta form or precystic form. Then the third one is the cystic form. Now coming to the life cycle of this Entamoeba histolytica, as I told you, this is a monogenetic. That means its life cycle is going to be completed only in one host, that is especially the man. So what is happening? So now the mature quadrinucleate cyst, that is the infective form to the man. Now when this mature quadrinucleated form is going to enter the human by the mode of infection through contaminated food and water, or by mechanically by houseflies, which fly on the contaminated food, okay, and then through uncleaned and contaminated fingers, even through the vegetables when infected with the fecus is used as a manure. So by all these means of root, this mode of infection is occurring by this mature cyst. Now what happens? Once they enter into the host, the cyst wall is resistant to gastric juices that are present in the stomach and cannot be destroyed in the stomach. So what happens? They will reach the cecum, and now each cyst liberates a single amoeba with 4 nuclei, which divides, and 8 daughter nuclei is going to be formed, with successive fission of cytoplasm called as amoebulae. And this process is called as excistation. So here you can see the mature cyst is going to converted into the amoeba stage, which we call them as excistation. That means the removal of the cyst from the cyst is going to be called as excistation. Now we are going to have each nuclei is going to act as each amoebulae. Now what is happening? Young actively motile amoebulae invade the tissues, so the liver, all the tissues, intestine, and lodge in submucous coat of large intestine. So here you can see them. They secrete the histolysine enzyme, which lyses tissue and lodge in the submucous coat of large intestine and multiply by binary fission. So now they are going to have the binary fission. They cause acute dysentery or ulcerative colitis by forming superficial ulcers with what we call as flask shape. I will show you them. Okay. Now, what happened? So here some of the trophozoites are going to again move or liberated into the lumen, where they transform into the cyst, and they are going to be um, encysted. That means they are going to form the encystation. So here the cell division, one, the trophozoite, where they will invade the tissues, and in other stage these trophozoites will undergo the encystation process and form the cyst, where they are liberated into the environment. When this cyst is going to be contaminated with the food, then again if they are going to be swallowed or going to be taken by some other host, that man is also going to have this amoebiasis disease. Okay, so this is the overall life cycle of the thing. So as I told you, some of the trophozoites are going to be liberated into the lumen, where they transform into the cyst through an intermediate precystic state. So we know that there is a minutiform intermediate also, and this whole process is called as encystation process. Now the cyst will be secreted into the fecus into the environment by patients or the carriers or the cyst passers. So other way that I told you is some trophozoites may escape from the intestinal ulcers into portal blood circulation and may be carried to liver, lungs, kidneys. So here you can see kidneys, even to the brain also, where they grow and multiply and cause extraintestinal amoebiasis but cannot form cysts. Okay, so those who are entering into or invading to the extraintestinal organs, they are not going to form the cystic form. They will be continuous in the trophozoite condition only. Okay, so this is the overall life cycle of Entamoeba histolytica. So this is what I told you, so you can have a glance of it. The quadrinucleate cyst, the mode of infection, then the cyst wall is going to be ruptured. It is going to be tolerated by the gastric juices that are present in it, and once they enter into the intestine, they are going to be liberating the 8 daughter nuclei, okay, where the excistation process occurs, and in which the trophozoites, some of them are going to invest the extra intestinal invasion, and some of them are going to form the again the cystic form, that is encystation occurs, and the cysts are liberated into the environment. So this is the overall life cycle that we discussed. You can have a glance of it. Then coming to the epidemiology, Entamoeba histolytica is worldwide in prevalence. So it is not confined to one region, it is worldwide in prevalence. And this is going to occupy the third most common cause of death from the parasitic diseases after schistosomiasis and malaria. And this amoebiasis disease is going to be found wherever sanitation is poor, and it is commonly occurs in the lumen of colon as a commensal causing asymptomatic infections too. And the transmission of this infection is affected through the ingestion of these cysts, that is the infective form of Entamoeba histolytica present in the contaminated food and water. Eating raw vegetables and fruits fertilized with the sewage can also be one of the reasons of getting this amoebiasis disease. And here the flies and cockroaches may act as a mechanical vectors in transmitting these disease, especially the cyst. So then moving to the pathogenicity and clinical features. So I told you some sort of a flask shape, all those things we'll discuss. The term amoebiasis is used clinically to denote all those conditions which are produced in the human host by the infection with Entamoeba histolytica at different areas of its invasion is going to be called as amoebiasis. And the amoebic dysentery term denotes a condition in which diarrhea and dysentery, most of the people are going to be confused between these two. So the term amoebic dysentery denotes a condition in which the infection is confined to intestine and is characterized by the passage of the blood and mucus in the feces, and it does not give the full clinical picture of the intestinal amoebiasis. Then how can we identify? The pathological lesions are of two types that are occurring due to this amoebic dysentery condition. The first one is intestinal amoebiasis or primary lesions. That means only we are going to see in the intestine condition. So this intestinal amoebiasis, so if you see the picture, here is an intestinal submucous region where the infection is confined only to the large intestine, where the parasite is going to produce some sort of a what we call it as a flask-shaped ulcers. So here you can see a flask-shaped ulcers which are superficial and extensive, okay, which are superficial, that means only upper layer you are going to have, which and extensive. Then moving to the second type is extraintestinal amoebiasis or secondary lesions. So here the trophozoites of Entamoeba histolytica, as I told you, sometimes carried into the portal blood circulation from intestinal ulcers from here and reach the liver, lungs, spleen, brain, and skin, etc. That means other than the intestine, they are going to extra-intestinal parts, where it produces the secondary lesions. The pus produced in the amoebic liver abscesses, so here you can see, is a chocolate brown color and is known as anchovy sauce pus. So here in this test tube, if you see here, we call it as anchovy pus, sauce pus. It consists of a dead liver cells, WBC and RBC. So amoebic liver abscesses, which is going to be caused by Entamoeba histolytica, serologically highly positive in extraintestinal cases. Okay, so this is all about the pathogenicity and clinical features of amoebiasis disease, which was caused by Entamoeba histolytica. Then moving to the laboratory diagnosis. So the diagnosis of intestinal amoebiasis is done by microscopic examination of feces and a saline and iodine mount, that means iodine preparations, iodine wet mount preparations to detect trophozoites and as well as the cystic stages, both. And the diagnosis of some extra intestinal amoebiasis, that is liver, brain, all those things, is done by microscopic examination of biopsy tissue of trophozoites. Biopsy means a little bit of that specific organ is going to be taken and biopsy is going to be done for trophozoites. And we are also going to have some serological tests to demonstrate the antibodies in patient serum by different methods like ELISA, RIA, latex agglutination test, counter immunoelectrophoresis, indirect heme agglutination, and even the complement fixation test. And presence of white stone-shaped charcot-laden crystals in the stool suggests the presence of Entamoeba histolytica. So these are the few laboratory diagnosis methods by which we can diagnose the presence of this Entamoeba histolytica in the patient's sample. Then coming to the prevention and control. Amoebiasis can be prevented and controlled by providing protected water supply, by providing improvement in sanitation and sewage so that water contamination is completely eliminated, then by providing or by avoiding the use of raw vegetables and fruits, and by detection and treatment of the carriers, and by practicing the personal cleanliness and maintaining the hygienic conditions. Then how you are going to maintain or practice the personal cleanliness means washing hands with soap and water after handling the dirty articles, before taking meals and after using toilet, and protection of foods and drinks from contamination by houseflies, cockroaches, etc., or by cutting fingers nails regularly, that means not growing of nails. Avoid passing out of stool on open ground, street sides, or vegetable fields, and also avoiding the use of unboiled water, improperly washed vegetables, and raw salad. So if you are going to maintain all those things, the personal cleanliness will be maintained. And the municipality also have to be responsible in providing the proper municipal hygiene, that is of supplying good protected water supply. All the things have to be done. That means covering of the food articles by the traders, chemical treatment of human feces to be used as a fertilizer, proper disposal of the sewage have to be done, proper sanitation of roads, streets, lanes, and open drains have to be done, purification of the water, of that means the drinking water have to be done. So if the municipality as well as the personal hygiene is going to be maintained, we can easily avoid this amoebiasis disease. Then treatment, different antibiotics are going to be used to control this one. Mainly the chloroquine and the metronidazole are the two drugs that are being especially used in controlling of this amoebiasis disease. So this is all about the amoebiasis disease. Thank you.